Bismillah ar-Rahman ar-Rahim. We start another paper 4 as exams are coming near and we need to revise these a little more thoroughly. So this is the June 21 paper 4-1 and this will be the first video on that. As you all know, the paper is for 2 hours and you have to be very careful. There will be, of course, there will be any more essay questions. So when you look at it, there's going to be here we have a section A and we have a section B, but there is no more going to be any section B. You are only going to have a section A. No more essay questions are going to come in the exam this year when you're going to take the exam next month. Now let's look at question number 1. Photosynthesis is a complex process involving the transfer of light energy into chemical energy. That's what we study in O levels. Describe the role of the photosynthetic pigments. Now what do the pigments do? So they absorb or trap the light or the photons, and then that results in the excitation of the electrons for cyclic and non-cyclic photophosphorylation, and the accessory pigments pass the energy to the primary pigment, and the pigments form a light harvesting cluster, or which is called photosystem, and you can have said PS1 or PS2. So for 2 marks you had 5 mark scheme points. Absorb light or photons, excite electrons, for cyclic non-cyclic photophosphorylation, accessory pigments pass the energy to the primary pigment, and pigments form a light harvesting cluster, photosystem 1 and photosystem 2. Then coming to the next part of the question, it says name the precise location in a chloroplast of photosynthetic pigments. Now that's of course and there's only 1 mark. I remember in one of the very old papers it was for 2 marks. So the mark scheme for 2 was thylakoid membrane and granum. But here they have allowed thylakoid. If you said thylakoid membrane, fine, that would have got you a mark. So thylakoid membrane would have got you a mark, or if you had said granum, you got you a mark for that as well. I said or, I didn't say both. So this would have got you the 1 mark. Then it says name a practical technique to separate photosynthetic pigments, and that's called chromatography. Chromatography. So that was just the name you wrote and you got a mark for that. Then we're going to go on to the next part of the question. It says the rate of photosynthesis is affected by many environmental factors. A student carried out an experiment to investigate the relationship between the concentration of carbon dioxide available to an aquatic plant, whatever, Kabamba, and its rate of photosynthesis. Figure 1.1 shows the experimental setup for this investigation. LED lamp at a fixed distance, sealed container, 15 centimeter length of that plant, aquatic plant, whatever the name was, and you have sodium hydrogen carbonate solution. So you have a liquid inside this. Whenever you're talking of a liquid, remember either 4 or 5, you must talk of the volume and you must talk of the concentration. What is the concentration? Like 1% or 2% or 3%. And then you see there's a LED lamp which is at a fix, and there is this scale here. So you can see, you can measure the distance of the scale. So you must take time like this and read all this and then continue with the next with the question on this part of this diagram. Let's now read it all very carefully and slowly. Sodium hydrogen carbonate solution was used as a source of carbon dioxide. Why? Because it's an aquatic plant. And if it was in a pond, it was in a pond weed. So in a pond, you would have a lot of microorganisms in the pond which would be producing carbon dioxide. You could be having fish in the pond and those could be producing carbon dioxide. But here you've used tap water. So in tap water is not going to have any carbon dioxide. So you have to add a chemical to it, and that chemical is sodium hydrogen carbonate. So the concentration of carbon dioxide was varied using 6 different concentrations of sodium hydrogen carbonate solution. 1%, 2%, 3%, 4%, or 5%. I mean, we can say it in anything. And as you can see here, they've given it to the concentration in mole dm cube. Okay. So the concentration of carbon dioxide was varied using 6 different concentrations of sodium hydrogen carbonate solution. and plants were kept in the dark before the light was switched on at the start of the experiment. So they were kept in the dark before the light was switched on. 5 replicates were carried out at each concentration. So whatever the concentration was, 0.25, 0.5, 0.75, 5 times the experiment was repeated. Replicate means repeated. The rate of photosynthesis was obtained by calculating the percentage change in dissolved oxygen concentration in the solution over 5 minutes. The rate of photosynthesis was calculated the percentage change in the dissolved oxygen concentration in the solution over 5 minutes. Let's look at the solution again. So there is a solution here inside it. And so what were we measuring? We were measuring the percentage change in dissolved oxygen concentration in the solution over 5 minutes, because as the plant was photosynthesizing, it was producing oxygen. So the oxygen concentration would change over 5 minutes. Figure 1.2 shows the results of this investigation. Now let's look at the result of this investigation. Now when you look at the results of the investigation, what do you have on the y-axis? Percentage change in dissolved oxygen. And this is minus 15, minus 10, minus 5, then 0, then plus 5, plus 10. Right? So it is increasing, and here on the x-axis we have concentration of sodium hydrogen carbonate solution in mole dm cube. So 0.0, 0.25, 0.50, 0.75, 1.0, and 1.25. So look at the graph carefully, read the graph. Now it says, with reference to figure 1.2, explain the pattern of results obtained between 0.25 mole dm cube. So here is the 0.5 mole dm cube and 1.5 mole dm cube, 1.25. So here, so what have we to explain is, we have to explain this part of the graph. We have to see how the oxygen, dissolved oxygen, is increasing, and then it's sort of becoming, it's plateauing. It's becoming constant. So the oxygen, percentage change in dissolved oxygen, yes, it's increasing up till here. But then as you can see here, it's become constant here. So it has become constant at 30. 30 is the 30% is the percentage change in dissolved oxygen. So it's not even the percentage, it's the percentage change in dissolved oxygen. So with reference to explain the pattern, now it says it's an explanation. It's not a describe, it's an explanation. So explain means you've got to give me a lot of bio here. So from 0.25 to 1, concentration is the limiting factor. From 0.25 to 1, let's look at it here. This is 0.25 to 1. You see how it is increasing. So it is increasing from 2.5 to 1. So you see this point here, it's increasing. It's increasing more than gradual, and it's increasing up to here. So 0.25 to 1, the concentration of carbon dioxide is the limiting factor. Whatever I've told you is on the x-axis, that is the limiting factor. So the concentration of carbon dioxide is the limiting factor. As we've increased it to 0.5, more photosynthesis, as more 0.75, more photosynthesis. So concentration of carbon dioxide is the limiting factor from 0.25 to 1 mole dm cube. Right? Then at more than 1, now carbon dioxide is no longer limiting. Then what are the other factors which are limiting? Now could be light intensity, could be temperature. You see, I always make this mnemonic TLC: temperature, light intensity, and CO2 concentration. So temperature, light intensity, and CO2 concentration. Now if CO2 is no longer, so what are the other two factors? Could be temperature, could be light intensity. So at more than 1, light intensity or temperature is the limiting factor. Okay, then explanation. Why? Why do we need carbon dioxide? Carbon dioxide is needed for the Calvin cycle. Why? Because it reacts with RuBP. Now coming to the part 2 of the question, the percentage change in dissolved oxygen by C whatever at 0.0 mole dm cube of sodium hydrogen carbonate solution is negative. Suggest reasons for this negative value. So why is the oxygen gone in the negative? The reason is that there's no photosynthesis. There is either little photosynthesis or there is no photosynthesis. And then what is oxygen being used for? Oxygen is being used for respiration. Oxygen is used in respiration, so that would get you your 2 out of 2. So because there is no photosynthesis, because oxygen was being produced by photosynthesis, but then because whatever oxygen was, it's going into the negative is because it's being used up for respiration. Then we come to the part 3 of the question. It says to minimize temperature changes, the student decided to use an LED lamp as a light source. LED lamps release very little heat energy. Explain the importance of minimizing temperature changes in the experiment. Naturally, why are we so worried about temperature changes? To control or to ensure that it's a fair test so that carbon dioxide is the only variable. Otherwise, we'll say, oh, the temperature is also changing and then the carbon dioxide concentration and all that. So we have to minimize the other things, other factors. Then low temperature means low kinetic energy, means low rate of reaction. Then low temperature decreases the enzyme substrate collisions. Then lower temperature is a limiting factor. Then higher temperatures increase in kinetic energy. Maybe will decrease the rate of high temperatures can decrease the rate of reaction of the photosynthesis, and high temperatures can also denature enzymes, and high temperatures may cause increased photorespiration, which of course you don't study anymore in the new syllabus. So explain the importance of minimizing the temperature changes in the experiment. You can go through all the points here, and you can see how what the points that I have been discussing. You can pause the video here and have a look at all this. This is the last point I said was to control a variable to ensure a fair test. Then I said low temperature kinetic energy decreases, so rate of reaction or photosynthesis decreases. Low temperature kinetic energy decreases, name enzyme substrate collisions will decrease. So enzyme substrate collisions will decrease. Then lower temperature is a limiting factor, limit the rate. Then higher temperatures increase in kinetic energy, decreases rate of reaction of photosynthesis or oxygen production, could result in denaturing the enzymes, and increase in temperature causes increase in photorespiration. Now, coming to question number 2, all organisms aspire to release energy from energy-rich molecules such as glucose. Anabolic reactions use energy. Anabolic. This is not anaerobic. State 2 examples of anabolic reactions occurring in living organisms. Well, of course, anabolic means to putting the monomer to a polymer, so DNA replication, or you can say transcription even, or to make up or build up a protein. So you could have said DNA, you could have said putting the nucleotides together to make, so it's DNA replication because it stated 2 examples. So if you said DNA replication, this would be a better answer than just saying putting the nucleotides together to make a DNA. So DNA replication, or if you said transcription, because in transcription what you do is you put the mRNA nucleotides together. So transcription, then to build up proteins. So putting the amino acids together to build up a protein, or even if you named a protein, like you said, to build up a hemoglobin molecule, we'd give you a mark for that. Then you could have said to build up lipids, triglycerides or a phospholipid, or you could have said to make up a carbohydrate, like you could have said glycogen, or you could have said starch in plants. So anything of that could have been correct, or if you could said phosphorylation, which is the formation of ATP. Then coming to part 2 of the question, describe and explain the use of ATP in glycolysis. Well, I'll just revise glycolysis with you in a minute, and then you can figure it out how the use of ATP in glycolysis. And please remember, this is 3 marks. Describe and explain the use of ATP. Now, if you look at this glycolysis, where do we need ATP? We need it here. We need it here, glucose to glucose 6-phosphate. Then glucose 6-phosphate becomes fructose 6-phosphate, but no ATP is needed here, so we don't need ATP here. Then fructose 6-phosphate, we need another ATP to make it fructose 1,6-bisphosphate. So glucose to glucose 6-phosphate, and then fructose 6-phosphate to fructose 1,6-bisphosphate. That's exactly what's in the mark scheme. Glucose to glucose phosphate, and the second one is glucose or fructose to fructose 1,6-bisphosphate. And why do we need this? Why need it? We need because you see what you have to understand is that the glucose has entered Glucose has entered through a channel protein, and then as soon as it's converted to glucose 6-phosphate, now it cannot go out of the cell because it becomes a large molecule, so it cannot go out of the cell. So glucose cannot leave the cell. So why have we converted into glucose 6-phosphate and fructose 1,6-bisphosphate? Is basically, of course, it's the series of reaction which is going to result in the formation of pyruvate, and you're going to get a net gain of 2 ATP. So, but we basically the purpose is so that glucose cannot leave the cell, and of course it activates or increases the energy of glucose, or it decreases the stability of glucose, so that then we can make out the NADH, and we can finally get it to pyruvate, and then the pyruvate can enter into the link reaction, and the Krebs cycle will take place. Now, coming to the B part of the question, some factory workers in the early 20th century were exposed to chemical X and experienced serious side effects. Some of the effects of exposure to chemical X are decreased production of ATP, increased lipid metabolism and weight loss, so your fats were being used up and you were losing weight, increased production of pyruvate and lactate. Whenever it says pyruvate, you know when is pyruvate made? It's made at the end of glycolysis. When is lactate made? At the end of anaerobic respiration. So excess heat energy release causing an increase in body temperature, which can be fatal. Chemical X increases the permeability of the inner mitochondrial membrane. So this is the mitochondria. This is the inner mitochondrial membrane. So it increases the permeability of the inner mitochondrial membrane to protons, which is hydrogen ions. So the hydrogen ions would pass back into the matrix and they would not pass through the ATP synthase. Chemical X increases the permeability of the inner membrane. Normally what you have ATP synthase here, and the hydrogen ions should pass through the ATP synthase and ATP should be made. But what if the hydrogen ions are just coming back into the matrix without passing through the ATP synthase, so there won't be any ATP being made. So chemical X increases the permeability of the inner mitochondrial membrane to protons, hydrogen ions, causing some protons to leak into the matrix. So always do a sort of a diagram and explain this to yourself so as to do paper 4 in a better manner. Part 1 on this question was explain why people exposed to chemical X show number 1 decreased production of ATP and number 2 increased lipid metabolism. So there are two different points, and look at the marks, there are 3 marks. So for decreased production of ATP there would be what? There is less steep a proton gradient. You see, this was the outer membrane and then this is the inner membrane. So if the hydrogen ions are coming back, then the hydrogen here is less. So there is a less steep proton gradient. Normally there would be sort of a lot of hydrogen here and there would be no hydrogens here. So there would be a steep gradient and this would come through the ATP synthase. Now because they are leaking back, so there is no proton gradient, and fewer protons go through the ATP synthase. So there will be decreased ATP production. Now for the next one, lipid metabolism. What are problems are we going to have? Why? Because more of the lipid are being respired to release energy. So more of the lipids being respired, or the body fat being respired, or the lipid stores being respired, and less ATP is made from glucose. So increased lipid metabolism because less ATP is being made from glucose. So there's going to be increased lipid metabolism. So for increased lipid metabolism, you would say respire body fat as less ATP made from glucose. Then come to the part 2 of the question. Suggest and explain why chemical X causes increased production of pyruvate and lactate. So why was chemical X causing increased production of pyruvate and lactate? Why? Because there was increased rate of glycolysis. You know, glycolysis is glucose to pyruvate, 1,6-carbon glucose to 2,3-carbon pyruvate. Why? Because the oxygen was being used faster, and so this resulted in increased anaerobic respiration. You had to reduce anaerobic respiration to lactate. Lactate is only produced when there's anaerobic respiration. And the last part is suggests why excess heat energy is released. Why heat energy is released? Energy from reduced NAD. Maybe energy from reduced NAD reduced NAD was not being converted to ATP, so reduced NAD released as heat. Energy from reduced NAD released as heat. Or what could be possible is energy not used to join ADP and PI. So the energy which was needed to join ADP plus inorganic phosphate was not being used, so released as heat. That finishes the 11 mark question, which is the second question on respiration. Now coming to question 3, in 2014, scientists published a phylogenetic evolutionary tree diagram showing how and when different groups of insects developed from ancestral groups. To construct the diagram, the scientists compared nucleotide and amino acid sequence data for 1478 genes from each of 103 different insect species. The 103 species represented all 32 orders of the class Insecta. The data were used to arrange the species in related groups according to their degree of molecular similarity and to track how sequences changed over time. Outline 3 ways in which the members of a species are similar. Very basic, is it? Asking you the definition of species. How are they similar? They are similar morphologically. Morphologically. So you could have said anything: morphologically, physiologically, biochemically, genetically, ecologically, behaviorally, and can interbreed to produce fertile offsprings. So any three of these would have got you your 3 marks. Then we come on to organisms are classified in a hierarchical system of taxonomic units. Name the domain and kingdom to which insects belong. Domain naturally is eukarya because you have 3 domains. What are the 3 domains? Archaea, bacteria, and eukarya, or archaea, prokaryotes, and eukarya. So this will be eukarya, and the kingdom will be Animalia. Then explain why a phylogenetic tree diagram is useful for classifying insect species. Now, why do we need such a tree or a diagram for classifying insect species? So basically, why do we draw this tree diagram? Is it helps to identify the larger taxonomic groups, and then taxonomy shows evolutionary relationships, and species that are closely related should be classified together. So a bit difficult to word it, but if you knew it, you could easily find some way to sort of write it in the correct biological English. Now coming to the B part of the question, for this study, new software for processing large quantities of data was developed. Calculate the total number of sequences that were compared, considering both the nucleotide sequences and the amino acid sequences for each gene. Now here, what do we see here? 1478 genes were being processed and 103 different species. And then we are doing the nucleotide and the amino acid sequence. So naturally we would do 1478 into 130 into 2, and this would be the answer for it. Then coming to the last part, explain why the study needs to make use of bioinformatic techniques. Why? Because it was a lot of data. It was a lot of data to store or analyze all this and large quantities of data, and to compare many sequences, and of course to calculate or measure the quantity or the percentage of similarity or differences. And it was fast and it was accurate and efficient. So all these factors are why the bioinformatics. That's a very typical question which they ask you on bioinformatics and all this database that they have introduced new in the syllabus. Basically, what does bioinformatics do? So a large quantity of data needs to be stored somewhere, and then it needs to be compared. Now, a computer can do that wonderfully. If we'd asked humans to do it, we'd need large number of people to do it and spend hours and hours processing it and comparing the different sequences. And then we could calculate the percentage similarity. How much of it is similar? Is it 70% similar? Is it 80% similar? Is it 90% similar or less? And then, of course, all this was very fast and accurate and efficient. We're a machine. We've made a machine. Human being, of course, have made this machine, this computer, which will beautifully do all their work for them. So it's an era of computers, and everybody should specialize in computers rather than doing anything else. We finish this here, the first 3 questions, and then we'll continue in the next video, the remaining questions. Quick revision of glycolysis: glucose to glucose 6-phosphate, glucose 6-phosphate to fructose 6-phosphate, fructose 1,6-bisphosphate breaks into triose phosphate. So 3 carbon is formed here, 2, 3 carbon formed here, and then 2 NADH formed here, then 2 ATP formed here, then nothing happens, and then there's intermediate steps, and then last 2 ATP made, and finally we have 3 carbon pyruvate, but they've only shown you one side of it. Similarly, this one becomes this, and this will go twice. So we have 2 pyruvates, so 3 carbon pyruvates. Please remember, I don't expect you to remember the other names, but I expect you to know this, where NADH is made, where the 2 ATP made here, and then the 2 ATP is made in the last stage just before pyruvate is made. So just a quick recap of glycolysis. Thank you, and thank you for watching.